Hello and welcome back to GCRC and Friends. We're in the middle of the COVID-19 pandemic, and we are slowly progressing towards the confinement and return to work. And as we do that, everyone is wondering whether it's safe to do so. And everyone is wondering as when we'll have actually drugs and remedies and vaccines to safely get back to our lives. So this is a question really of the pharmacological process, the discovery of drugs and the development of treatment and a vaccine. And today we invite a friend of mine and a really well-respected researcher at McGill working on in the Department of Pharmacology to discuss this and the process of development of therapies. And so welcome Thierry Hébert for joining. Terry is a professor in the Department of Pharmacology and Therapeutics at McGill. He's involved in everything in the community at McGill, but also in broader sense. And he's also one of the teachers who cares a lot about his teaching and uses social media and multiple platforms actually to engage his students. So it's something that I've picked up from Terry from the beginning. He keeps his students interested and he keeps educating them through multiple platforms. So I felt that it was a wonderful fit for Terry to come and talk to us about the COVID-19 drug development process. So thank you very much, Terry, for joining today.
Thank you, Thomas, for inviting me. I'm very happy to be here. I'm happy to kind of discuss where we're going with COVID-19 therapeutics, and I would like to thank you for thinking of me.
My pleasure. So I believe you will introduce us to your beautiful lab there. As PIs, very often as professors at university, we're seen essentially as teachers, but we also mentor community and who are actually the people who carry out the research in our lab. So I think it's really important to introduce our labs like this. So I believe that's what you'll introduce next, right?
Exactly. So I have what I think of as an amazing group. Here's a mixture of my current students and some students who just recently graduated. They're a mixture of undergraduates, graduate students, and postdocs, and I also have 4 really important research personnel who've been with me for, some of them have been with me for almost 20 years. We work on drug discovery on a class of targets called G protein coupled receptors, which are literally involved in almost all physiological processes, and maybe as we go forward, we'll touch a bit on that in the context of COVID-19. But I think that as Thomas says, it's important to thank the people who actually do the work in our labs. It's important to give them their due in their time in the sun, because at the end of the day, they're the ones who do the experiments that allow us old guys to write up our papers and make the field move forward.
Yeah, they're also the discoveries of the future, right?
Exactly.
All right.
All right. So we're going to talk today about some of the prospects for treatment of COVID-19, and you may have heard this virus called SARS-CoV-2. Or COVID-19. COVID-19 sort of refers to the interaction between the virus and the host, right? COVID-19 is the disease that's caused by SARS-CoV-2. It's really an interaction between the virus, pictured here, which is really a fairly simple sort of biological entity. It has a coat which has a bunch of spike proteins which allow it to interact with host cells. And that coat surrounds the RNA that's inside, the nucleic acid that the coat protects. So it's really a very simple, if you want to think of it as a life form, but it can do a lot of damage because it's almost purpose-built to infect cells and co-opt their own physiological processes and make people sick.
Yeah, so we're going to talk. Yeah, Terry, I think it's important maybe to mention that we saw quite a bit of the biology of this virus with Selena Sagan from in our previous episode of this series. So it might be a good watch essentially to get into the molecular biology details. But one thing that you mentioned that is very, very important for the whole presentation today is that the disease in an interaction between the host and the pathogen, the virus. It's not something that is proper just to the host or just to the pathogen. It's really interaction of both. And I think the pharmacology and the clinical trials and the drug strategies that you'll present today reflect that.
Yes, they reflect how to interfere with host processes that might be critical for the virus to reproduce, and they also reflect the desire or our ability to interfere with the processes that the virus uses itself. So we'll be talking about a combination of both of those. And Thomas, thank you for reminding me about Selena's talk, because it keeps me from going into areas in which I know very little about. Thank you. Okay. So, we hear this almost every day. Is COVID-19 different than the seasonal flu? I think there's just two things that I want to mention with respect to that. It does have symptoms that are like the seasonal flu, so it's I suppose easy to get them confused. But the thing that we see more with COVID is that it has two features. One is that it can make people a lot sicker than the flu, although it doesn't always. And people who get COVID-19 are more likely to infect other people. So you'll see this number called R0. That is what we call the reproductive number of a virus. For the flu, it's something like 1. That means every person who gets infected will infect one other person. But with COVID-19, with SARS-CoV-2, typically 2 to 4 people can get infected. So you can see that a lot more people can get the disease by basic contacts between people, which is why we're sort of in this lockdown phase, trying to reduce the number of people who are getting the disease at any one time, so that our healthcare system is not overwhelmed. I think we're going to see when we reopen that people will get sick again, and we might have to go through waves of this happening until everyone gets the disease and we develop some sort of immunity to it. But we have to. What we've been doing is buying ourselves time to identify or discover drugs that can be used to treat the disease, to make the time in hospital, the time sick less. And it's also given us time to kind of prepare strategies to develop vaccines, right? So what all of these things do is not just prevent the disease from spreading, but they also preserve our ability to treat people who get sick, and they also buy us time to develop therapies and cures, hopefully.
Yeah, so we'll discuss quite a bit about that, whether a strategy is adopted to prevent infection, i.e. Vaccines, treat the early phase or treat the later phase. And there are different treatments, different strategies for each of them.
Yeah, and I'll just say that the rest of this slide kind of shows that there is a period of time when people don't seem to be sick, right? And yet they're still able to transmit the disease. So there's different phases of the disease. Once you become exposed, right, there's a certain time before you become infectious, and then there's a certain window between the time you become infectious and whether or not you show symptoms, right? So the idea is that even if you're not sick, you can transmit this disease. So it is something that we worry about, because people who aren't sick, don't feel like they're going to be able to transmit the disease, but that's not the case. So most people recover. Some people it takes a very short time. Some people it takes a longer time, and so, but we can talk about that as we go along.
Yeah. There's a bit more complexity also, which is not reflected in all slides, and I think we'll mention a little bit the differences in the population between the age groups. For example, has been really, really important in the progress of this disease, and also the strategy to treat will be different, and maybe the drugs that we'll use will end up being different depending on which part of the society, which part of the patients we're talking about. The difference between men, women, and there's difference between age groups as well, but also other differences like people that have pre-existing conditions, for example.
And that's also what the lockdown has kind of given us time to appreciate, is that the disease is different in different groups of people, and understanding how it's different has given us ways to treat the disease with the tools we have now, with just regular medical care, without even drugs, just being able to support people as as they're sick and prevent them from becoming more sick, and so that they recover. As Thomas says, the disease is different in different people. So it's something that we've now learned since March when we locked down, and there's been I don't know, thousands of studies now that have come out that have talked about the different ways that the virus affects different groups of people and how to deal with that. Right. So there is a spectrum of illness severity. That means that you can essentially go from having a mild version of the disease, which will feel like the flu or some mild form of pneumonia. That's what happens in most people. At least 80% of people so far have shown this type of disease. You can usually expect to recover pretty quickly from that. There's even some that don't show any symptoms at all, right? But they're still contagious, and they could still transmit the disease to people who are more susceptible to the more severe forms, right? So that means you have trouble breathing. That means that's what dyspnea is. You become hypoxic. That means your tissues don't get enough oxygen. 14% of people who get that disease, who get this disease, show some sort of lung involvement, in the sense that they have trouble breathing. And if you have trouble breathing, that's a clear indication that you need to be hospitalized. And in a smaller percentage of people, the disease becomes critical. So respiratory failure, shock, multi-organ dysfunction. The more we learn about the virus itself, the more we realize is that the virus affects many different types of tissues and organs. It causes damage in those organs. There's a component to the disease that may involve the blood clotting system, which may explain a lot of the severe disease because of multi-organ failure, because the clotting defects, or it may cause heart disease because of that, right? So as Thomas said earlier, it's really a different disease depending on how old you are, on your gender, on your underlying health conditions. It's really kind of, we've had time now to appreciate the spectrum of illness severity.
Yeah.
Before December 2019, no human anyway, had ever had this disease. So we've had to start essentially from nothing to learn about it.
Yeah, it progressed extremely fast, right? It was scary. It's been scary all along, but it was quite interesting to see how fast we learned about that disease. One feature of COVID-19 was just how broad infection, the cell types that are infected in the organism can be impacted. The receptor is expressed in multiple cells, and I think, as you mentioned, there's a much broader range of impacts on organs than just the lung, which was the focus of attention at the beginning because of essentially the disease. And as you mentioned, there's a lot of it relates to the immune system, or the systemic inflammation associated with the infection, whether, for example, in the kids having the discoloration of toes, all the way to the blood clot that you're mentioning. I think it's something that was underappreciated at the beginning, that is emerging and is probably in the broader range of the impacts of the disease.
Yeah, I've been reading a lot of books about pandemics and about the history of pandemics, and it follows a similar track, right? And in the sense that there was so little we knew about it in terms of its symptoms, in terms of its contagiousness, and all of which we found out in the last 3 months.
Yeah,
it really says, what we can do as a society when we face a problem, and that we actually try to deal with it, as opposed to plan for it not to happen, which is kind of what we have been doing in the past. So,
yeah, very inspiring to see smart people and dedicated people come together planet-wide to find a solution to this. I agree. I think it's spectacular. We could have done better, though.
Yeah, we didn't prepare for it, right? That's what we've come to realize, and you can argue up and down about different countries and their responses to the pandemic. No Western government has done really well. You can talk about we're still not testing enough people. We're still not functioning in Canada with a healthcare system that is effectively coordinated between the federal and provincial health ministries, right? I mean, you still see a lot of human weakness driving this, turf protecting, infighting, all of it, right? We as you said, Thomas, we could do better. but
Yeah,
it could have been worse too.
you're right. Yeah.
Okay, so now I guess we're going to talk about it from the perspective of someone who thinks about drug discovery, someone who thinks about pharmacological treatments for the disease. So one thing that the lockdown has given us time to do is kind of develop a more comprehensive understanding of how the virus gets into the host, hijacks host machinery to reproduce itself during the course of the infection. And so now, once we've learned a few of these things, we need to apply this knowledge toward either developing new drugs, which is probably the type of drug discovery most people are familiar with, right? But we can also think about what we call repurposing old ones. And as you'll see in the next couple of slides, the difference between developing a new drug and repurposing an old one is really using an old drug for a new purpose. It takes a lot less time than developing a new drug from scratch. So the hope now, as we wait for a vaccine, is that maybe there are drugs already out there that are not designed to deal with SARS-CoV-2, but may actually be able to be used to treat people who are infected with this virus.
Yeah, I think it's going to be the recurrent point. We're going to come back in this presentation, really, how the repurposing of drugs that have been validated and tested, in matter of reducing the risk also will be addressed quite a bit, but how this is much faster than developing something new or modifying a previous drug and having to rerun it through clinical trials.
And if we think like a drug company for a second, faster also means necessarily less expensive, which is also a good thing from that perspective too, right? I mean, the sooner we can get drugs out, the fewer regulatory hurdles we have to jump, the better, right? And that's what we need until we have a vaccine or until we have a community immunity. Is we need drugs that can treat people who get sick until we can cure the disease.
Yeah, and that time window can be mounted to a number of human lives saved.
Yes.
And in pandemic, we're talking about thousands and thousands of lives. So that speed is critical.
So if you think about how drugs are discovered like from scratch, one thing that Thomas and I will probably agree on is that there's a lot of work that's done in labs like his or mine that never gets any credit for by drug companies because most of that research is publicly funded through research agencies in Canada or in the provinces, where we do studies in vitro, in cell models, where we kind of we learn to understand a biological process, and then we make molecules that can impact that biological process. And so that process might be, a disease. And so the idea would be that, okay, you then go on to an animal model of the disease where you test and see if your drug candidate works, is safe, and then you try to understand how it works, right? And that can take years. So just to get to the stage where pharma takes it over and starts to do trials in patients, right? So the first phase of trials in humans is just to see if the drug is safe and to understand how the body deals with it, right? How the body metabolizes it, how the body distributes it, how it's excreted, et cetera, how long it lasts in the body. We call that process pharmacokinetics. Then there's a next phase where we actually look in patients who have that disease, a small sets of patients where we're actually looking to see if the drug impacts the course of the disease. And if that's true and the drug is still safe, then it will go to these larger trials called phase 3, where you're looking at a larger number of patients and you're blinded such that the investigators don't know who got the drug or who got a placebo, so they have a control to understand whether or not the drug actually worked better than the standard of care for that disease now. And all through that process, you have studies that will examine how the drug is metabolized in humans and how safe it is, right? So that process, again, can take years to complete. And trials are more and more complicated now. They often involve multiple centers in multiple countries with multiple regulatory frameworks that all have to be integrated. And that can take years to do.
So in a nutshell, this kind of time frame is scary for anyone with the disease or with their loved one who are sick in the middle of pandemic. It just cannot be applied realistically in the middle of pandemic.
No, and well, it can, but it won't help the people who are suffering now,
That's it.
right?
Yeah.
So, and maybe a lot of people don't realize that there's a fourth phase to drug trials, which is like, let's say 10,000 patients are tested in the trials before the drug is sold, before it goes onto the market. Then it's up to doctors to examine in millions of patients over the years whether or not the drug really works, to identify side effects that were not identified here, to identify adverse consequences that were not identified here. It really puts the onus on the physician, on your family doctor, in a sense. So every time you go to the doctor and a drug you take makes you feel bad, you ask your doctor, have you reported back in post-marketing surveillance to right? So that process also before a drug, I mean, will inform whether or not a drug goes back to the drawing board to make a better version of that drug. And so it can take between 16 and 20 years to get a drug out to the market. And the patent really only lasts for 20 years, right? So drug companies are always looking for the next best thing, which is fair because there are commercial entities. But just to say that this is what we're up against if we're trying to develop a drug from scratch to treat COVID-19. It will take years to get us to that place.
Yeah.
So what we've been thinking about then is, are there drugs out there now that we could use that won't be perfect drugs, but that will slow down the progression of the disease, that will make the course of the disease less, make the person less sick or make them get better more rapidly? And that's what we think of in terms of repurposing a drug. And if you think about it, those drugs have already gone through the drug discovery process. They've already been tested preclinically. They've already been tested in phase 1, phase 2 for that. And so you would save not only a lot of time in repurposing a drug, but you would also save a lot of money in terms of the $1 to $2 billion dollars it would cost for a new drug, the 12 to 16 to 20 years. We can actually reposition a drug or repurpose a drug for a lot less money and a lot less time. Now, this is sort of a pre-pandemic timescale. The timescale we're working on now is more like months rather than years, right? So we have drugs that we've built for other viruses or for other indications where we now know that they can impact the course of COVID-19. So this is where we can probably invest most of our time and energy in developing therapeutics that will give us more time to make a vaccine, or to make or for people to become immune by just having the disease and developing their own antibodies. That's what repurposing is designed, or is thought to hoped to give us time to do.
Yeah. But even when you look at this time frame of 6 years, you really see the contrast between what is mentioned now and what people expect or want essentially in the middle of pandemic, which brings the factor essentially that is central to the situation now, which is the risk factor. Is the risk factor evaluated under a different light in the middle of pandemic? And are we willing to go faster at the expense of risking the health of people, with even if it's a drug is repurposed and was used before, we're probably not going to go through as much of a testing before we administer to a patient, for example. And this risk factor can be reweighted and can be modified, I think, and the scale can be changed depending on things like even political pressure in the end. So it's something that we really need to be careful and critical about, I believe.
This disease has already killed hundreds of thousands of people worldwide, infected millions of people, but it's not the worst disease we could ever see, right? I mean, we're sort of I won't say dodging a bullet, but there are diseases out there that could be a lot worse than COVID-19. So the process we develop here is one that we should think about developing for things going forward as well, right? There has to be a plan put into place that allows us to shorten both of these timescales going forward, right? So I'm not sure how to shorten the de novo drug discovery process, but I'm thinking that there has to be some way to incentivize pharma to do this more quickly, right? Governments have to become involved at some point. to make that happen. Maybe you could say that, all right, why don't you invest in pandemic preparation, and we'll extend the patents on some of your blockbuster drugs like statins or whatever, right? So you'll make more money from your old group of drugs, and you plow half those profits back into, figuring out how to shorten the drug discovery process for pandemic diseases and preparing for the next ones, right? You could make the same argument about our need for new antibiotic drugs to treat bacterial disease.
Absolutely, yeah, because this is coming also.
How do we incentivize pharma to kind of still make money, but if corporations want to be treated like citizens, they have to be having some of the responsibilities of citizens, right? So
I believe
they owe us something.
I believe many companies now have this in their mind. We need to come up with a winning combination, right, for everyone. I guess everyone will be thinking about that in the.
Well, I hope so. I mean, like we as a species, we don't seem to be able to plan, right? So I don't know if that's a genetic defect or just part of our desire for good outcomes is to hope for them without actually planning for the worst. But
we'll also forget.
Yes, that's it. If you think about it, like our grandparents grew up in a world, or were born into a world that did not have antibiotic drugs, right? Like half the children born died, but we take antibiotics for granted. We take vaccines for granted, and so now you have people who don't remember that, who are anti-vaccine, or who don't really appreciate what a post-antibiotic world will look like. Those of us who think about those things are actually quite frightened by what we see, right? We need to kind of remind people all the time about what the world was like before vaccines and before antibiotics.
Yeah. And there you go, the importance of education, of being scientifically literate. I think this speaks to the core of what I believe in as a scientist, but also an educator, and I'm sure it's the same for you and for most of the colleagues, if not all the colleagues.
It's exactly the same, right? I mean, we as university professors, we're supposed to teach students, the material they need to adapt to become into their professions. But I think we have a broader duty to make them into informed citizens, right? Not like this goes beyond just teaching science students to become scientists. It means teaching art students to think critically like scientists. It means getting everyone to have a set of thinking skills that allows them to function in a democracy, right? I mean, if we don't do that, we're not doing a service. We get bad politicians. We get people who don't have our best interests at heart, right?
Yeah.
Who are willing to lie.
A lot of it also is about being, you're talking about the importance of being good citizens, but scientific citizens. But I think a lot of it is communications and being willing to engage, right? Which takes a bit of courage in knowing how much of an arena now social media can be. So providing the toolkit to our trainees and the future scientists into how to engage advantageously and critically into social media and be essentially the beacons of light out there.
Yeah, and it also means that we have to adopt a position of humility before how much we don't actually know, right?
Yeah,
I see a lot of university press releases that sound like they have cured a disease based on one paper published by someone like me or someone like you. That isn't really how it works, right? Universities hype these things and they create expectations in the public that we can never meet, right? So we have a duty to kind of to be honest about what our studies show, and to be honest about how much we don't know, and to be honest about how the process actually works, right? Don't believe our own hype.
Yep.
Okay. Sorry, that was a digression.
It's a meaningful one though.
Yeah. So where are we now with respect to COVID-19? Like there is a lot of work ongoing to kind of develop different strategic or different therapeutic strategies, right? So you will be perhaps not surprised to realize that there is a lot of work being done to generate a vaccine. The problem with a vaccine is over the history of vaccines, the most rapid one to come into play, it took 4 years, right? So we need something before the vaccines come on stream. And so we might think about therapeutic antibodies. Again, that's like either using antibodies from patients who recovered from the disease, who are now producing it, or building antibodies that target proteins on the virus that we can use to prevent it from infecting cells.
So instead of preventing, blocking the infection, having an active role in the infection.
Reducing the contact of the viral particle with the cells that it would infect by hiding them, by hiding the parts that interact with cells, essentially, the way antibodies work normally. And so there's a lot of work being done also. And this is where I think we were putting most of our money into, well, not me, but like developing drugs that actually impact the virus's ability to reproduce, right? Its ability to use the cell to make new copies of itself. So antiviral drugs, that's to me where we should be investing a lot of our efforts until we have therapeutic antibodies or until we have a vaccine. And I will talk about others that might be important as well, because as Thomas and I have both said it in different points during this chat, is that the disease is really a combination of what the virus does and how your body reacts to the virus, right? So a lot of things that we might target are things like in the immune response. Like there are different phases of the virus infection of the disease where you want to increase the immune response. And then there's another phase where you want to decrease the immune response because people get sicker if you don't do that. So the idea is to understand how the virus does this and then use the drugs that will best be able to manage that accordingly. So there are other things going on. We talked in the last slide about repurposing. So this is just this is what it's written here as a scan to repurpose. That means they're looking at the whole, like drug companies are looking at their whole catalogs of molecules that they have, right? So to understand, do some of our existing medicines work against the virus? And so this number here is small. It says 12, but really it's 12 times however many number of drugs there are in their collections, right? So lots of different ways to get at a treatment for the disease that will buy us time until we have a vaccine to the disease.
Yeah, I think it's important to remind people that the vaccines, even though they are, they're the patient's own immune system's response to an antigen, it still needs to be tested in clinical trials. So it still requires quite a bit of time in order to know if it's safe or not.
I was going to say, like many of the medical students that I teach are talking about doing things like volunteering to have challenge trials for vaccines, where they wouldn't know whether they got the active virus to test whether or not a vaccine candidate works or not. They could be getting the virus with getting the control for the vaccine, meaning they're going to get sick. So that's a way to speed up the trials of vaccines. But until we have some way to treat the disease effectively, it is ethically wrong to challenge healthy people with this virus before we have a way to impact whether or not they're going to get really sick from the virus, right?
Yeah.
So we can't even use the fastest ways to develop vaccines yet because we don't have an effective treatment yet. Once we do, then we will also shorten the time of vaccine development, because even if they get the active virus and the placebo vaccine and they get sick, we can still make them better. But until we can make them better, we can't use the most rapid way to develop a vaccine.
And test it, yeah.
All right. So where can we attack the virus? Over millions of billions of years of evolution, viruses have basically, they're very simple, but they can do a lot of damage because they've evolved to kind of take advantage of things that in our cells that they can use, right? They don't carry a lot of stuff around with them. They just basically, they're looters. They take what they want where they want, right? So where can we impact the virus? So I'm going to take you through this slide a bit slowly. So this is what the virus looks like. To get into the cell, the virus has to bind to a receptor on the cell. That means that there's something on the cell that recognizes something on the virus and connects them together. And then, then the virus kind of, the coat of the virus fuses with the membrane of the cell, and that brings that whole viral particle inside the cell. And so now it's a virus surrounded by a membrane coat from the host. And for the virus to kind of reproduce, it has to uncoat. So it has to get rid of that membrane, and it has to open up, and it has to release its contents, which is a long molecule of RNA. And what happens then is that it co-opts the systems in our bodies, in our cells, to turn this RNA into a very long protein called a polypeptide. And one of the proteins that and what happens first is then that there are proteases in our body which chop that big long protein up to make the non-structural protein, so the parts of the virus that are involved in its reproduction. So one of the proteins that they make is this enzyme called RNA-dependent RNA polymerase. This is the viral molecule that allows it to reproduce, right? So it makes more copies of that RNA, and then other copies of that RNA are then translated back into the structural proteins that allow it to build a new coat, and then that allow it again to fuse with the cell membrane and get out. So one virus might get in, but 1,000 viruses might get out, because the virus kind of takes over the cell.
Yeah, the amplification step there through the RDRP is a critical one, and there's already quite a bit of therapeutic and development of drugs targeting this. I think not only for SARS, for the coronavirus, SARS-CoV-2, but also for other viruses out there. It's a typical target.
And that, as you say, Thomas, is also the basis for how we're repurposing some of those drugs developed for other viruses to be used to treat COVID-19, right? So if you think about now what I told you about that life cycle, so we're not even back to the host immune response yet, but so we could target how the virus interacts with its cell receptor, right? And that's one of the main ones that we hope will work. We could target how the virus uncoats. So that was the hope for a drug called hydroxychloroquine, which is an anti-malarial drug also used to treat lupus, that the President of the United States was pretty sure was going to work. It turns out that it doesn't work. It may actually do more harm than good, because it is a molecule that impacts a host process in a sense, right? So this membrane fusion and endocytosis is bringing in of cell membrane. It's a process that your body normally uses for a lot of other things, right? So it's not targeting a viral process per se. It's blocking the entry of the virus, yes, but it's also blocking a lot of processes that are going on in your cell normally. So it has a lot of adverse consequences.
So there's a fundamental difference there between the drugs that affect the host mechanisms and the drugs that really target proteins and genes or processes that are specific to the virus itself.
Yeah. Now, I'm not saying that targeting the host processes is bad, because as we'll see, if we're going to dampen some of how your body reacts to being infected, that those are host processes, and we'll come back to that in a bit. But so we could think about how to target viral attachment, viral entry, viral uncoating, and then we could think about all those core processes that the virus undergoes. So we could use enzymes that inhibit the proteases that cleave that long polypeptide into the functional proteins of the virus. We could use drugs that inhibit the polymerase itself, that prevent the virus from making more RNA to fill new virus particles, right?
Maybe remdesivir is the most famous one here, right?
Yeah, so remdesivir, it is not that old of a drug. It was designed as a hepatitis C drug. It didn't work against hepatitis C, but we found that it did work against the Ebola virus, right? So this is a drug that was made recently by a drug company called Gilead, or Gilead, I never know how to pronounce it, but it does show antiviral activity against SARS-CoV-2, right? It doesn't seem to stop people from getting sick, but what it does seem to do is reduce the time that people are sick, right? So to me, that's a that's an important advance. If you're
Definitely a
getting
win.
better more quickly, that's a good thing. So, and it's the same with these other drugs, these protease inhibitors. They are often purpose built for other viruses too, like HIV, for example. These other ones up here, these are more. This drug was probably developed for another SARS-CoV, which was CoV-1, the one that should have warned us that SARS-CoV-2 was coming, right? The near pandemic we had 15 years ago.
Yeah, we have MERS. We had MERS and SARS before,
and
right? And there were coronavirus diseases as well.
There have been canaries in the coal mine warning us, right?
Yeah.
But none have made us as sick as SARS-CoV-2, right? So the idea is that there are multiple places where we could attack the virus, either with drugs we already have, or with drugs that we can build with the specific purpose of blocking viral processes or the interaction of the virus with host cells. And the last thing I'll mention in this slide is that, so we know that there is an immune response in the host against the virus. It seems like at the beginning, when the virus is just taking hold in those cells, you would like to upregulate the immune system and increase the immune response. That would prevent some of this from happening. But once the virus gets out, then there's an interaction between the virus and the host, which actually causes sustained immune signaling, which is actually damaging to the host, right? So that involves a molecule called a cytokine, and this is interleukin 6. It binds to a receptor, and it really massively ratchets up the host immune response. And if it's not controlled, it can actually make people sicker. So
actually this uncontrolled or storm, if you will, of immunity is probably what's responsible ultimately to most of the damage that happens in patients.
Yeah, for patients who go into hypotonic shock or whatever, it's usually not because of the virus. It's because of what Thomas called a cytokine storm, where there's this massive uncontrolled inflammatory response that causes the cells to have damage where you get massive damage to cells, and you get a massive reduction in blood pressure, and people die because of going into shock, right? Their hearts no longer can pump enough blood to meet demand. So we were starting to think that maybe we could control this late aspect of the interaction between the virus and the immune system by inhibiting interleukin 6 signaling. And so there's been a lot of efforts to develop blockers or to repurpose known blockers of the interleukin 6 receptor that would reduce the possibility of cytokine storm, right? And so the host immune response would not be making things worse in this case. Okay, so that brings us to the question of what our actual goals should be, right? So there's three things that we could talk about, and you've heard of all three of them: testing, treating, and preventing. We've been talking mostly about developing treatments that would allow us time to be better at doing this, which is preventing. But we also need, if you think about it in both the short and the long term, we need better tests. We need to be able to follow people who are asymptomatic. We need to be able to follow people after they've gotten over the disease to see, how long that the immunity they get from having the disease lasts, et cetera, et cetera. And so a lot of our colleagues are working on developing these sort of molecular diagnostics to kind of build tests that can go out and that can go out and be used. But I think that we all need to encourage our governments, local, provincial, and federal, to kind of do more testing. And that's important both in the short term for our own pandemic, but also in the longer term to develop simple ways that people can use tests under conditions where there's a lot less resources. So poor countries, et cetera, places where there aren't large hospital academic research centers that can manage these tests, right? We need
it's important to keep that in mind. Not everyone has the same capacity to detect.
Right. yeah, to track the disease. And then the idea would be that ultimately we'd like to be able to prevent the disease, right? So we need to understand what immunity people get from having the disease and getting better, but we also need to be able to profit from that by using serum from those people to kind of treat other people who might have the disease or develop antibodies that we can use therapeutically. And then, of course, we need to invest the time and the money in developing vaccines. Lots of people are doing that, but it's going to take time, right? Thinking about it, just having the vaccine still means we have to test it, still means we have to manufacture it in billions of doses, and still means ultimately that we have to be able to deliver it in low resource settings, right?
Yeah, so the scale and the delivery are also challenges of their own. Even if you had the perfect vaccine, it wouldn't be tomorrow you would see it at your door.
No, so it's a question of buying ourselves time for this to happen. And by being able to treat the disease, by either repurposing drugs or more quickly developing drugs that are new that could be used, I think that's where we are now, right? So if I were to say where's our best hope for this, it's going to be in repurposed drugs that we have now that drug companies already know how to make at scale, that can be delivered in the number of doses needed. And down the road again, we still have to think about delivering these drugs. How do we make such drugs available in countries without reasonably large healthcare budgets, right? We're lucky in Canada. We never really take account of that very much. Our healthcare system isn't perfect, but it's still pretty good.
Yeah.
Okay. So if I had to pick one target that would be a good thing for a new drug, that would be like the best where I would put my money if I were an investor. It would be again, it's sort of kind of like repurposing already in the sense that for the first SARS that we saw back in 2002, we know what the receptor on the cells are, and so there's been a lot of development of a molecule that could impact the interaction of SARS-CoV with this receptor. And for good or for bad, the receptor on SARS-CoV-1 is the same receptor that binds SARS-CoV-2. It's a protein that's on a lot of different cells in our body called ACE2, or angiotensin converting enzyme 2. And so we know that by targeting this protein, and there are studies that show that this will work, that we can impact, we can repurpose any drugs we develop for this to be used on this. And then we also know that for the SARS-CoV virus that we saw in 2002, there is a protein on the that is involved in helping the virus get into the cells. And we've already developed an inhibitor there, and it looks like that protein is also on SARS-CoV-2. So we were thinking that you could either develop an antibody that would block this interaction, or some sort of decoy that would prevent the SARS from being able to interact with the host cell. It's gonna take time still to do that, but I just thought I would kind of tell you a bit about where that's going to go and why we still need other things in the meantime. But this is going to be one of the strategies I think that will work ultimately best.
Yeah. So the reasoning here is that maybe if there's been more than one coronavirus, having this as receptor, there will be more. And therefore, if we're to develop ways to block this interaction, then we might be able to reuse them again in the future for new viruses.
Exactly. The receptor is the same for the older SARS virus and for SARS-CoV-2. So there's already been some drug development there, but you can imagine that we stopped doing it once SARS went away, right?
Yeah.
So, but now it's back. So I guess this is a detailed slide that I won't spend too much time on, but one of the real interesting developments over the last 20 years in the field that Thomas and I work in is that we can now get the structures of these large proteins in many different ways at high resolution. And so this is the structure of ACE2, the receptor for the virus. And this is actually the part of the virus that interacts with the host protein. So we know a lot about that interaction structurally. So what that means in terms of drug discovery is that because we understand the structural basis of the interaction, all of the different nooks and crannies that they fit into one another, we can develop drugs that block that interaction, that prevent that interaction. So I'll just show you this one little slide, which shows that. So now this is the SARS protein that called the spike, which is actually what binds to ACE2. And you can see that when they simulate the molecular motions of that molecule, it opens up. It opens up to bind ACE2. And if we can figure out a molecule that would sit in that opening, it would prevent it from interacting with ACE2. So the idea would be that, so if this is the viral particle, under normal circumstances, the spike protein would allow it to interact with ACE2 here, and get into the cell and do its damage. So if the receptor here and the virus is here, then one way to prevent the virus from interacting with the cell would be to develop these therapeutic antibodies which actually bind the virus and prevent it from interacting with the host cell, right? Yeah. So the idea of therapeutic antibodies is that they essentially coat the virus, making it harder for the virus to interact with the host cells. So what would happen then would be the virus would just go out the way it came in, like it would go out and basically be excreted in feces and urine, right? It would never have a chance to infect cells. The other strategy would be to take this receptor and through genetic engineering, build a version of it that is not anchored to the cell membrane. So you would make what we would call a receptor decoy here, right? So the receptor then would do the same thing as the receptor on a cell or that antibody, in that it would cover the interaction site on the virus, again, preventing it from interacting with the host cell.
I think for both of these strategies, a lot of it is related to how we understand what recovered patients' administration of serum was. I think you could, there's been early studies, and I think it's still a strategy used in many viral infection where a patient that recovered from an infection can donate blood, you take the serum from this, and this can be administered. So part of it is that these antibodies could actually passively go and block the interaction between the virus and the receptor, but also stimulate the immune system of a different patient. But sometimes it works, sometimes it doesn't. Whereas in this case, it's more precisely engineered to do just that.
And we can also, instead of using antibodies from patients, we could actually engineer the antibodies ourselves too, yeah. Right? That would either do what the receptor decoys do, just prevent the interaction, or as you say, Thomas, that actually toggle the immune system as well, right? That bring more of the immune system to bear on new virus that is seen by a patient. But
this is something exciting. I think protein engineering and engineering antibodies is extremely exciting at this moment.
Yeah.
I think.
And the Nobel Prize Committee agrees with you, right? So that's it. So, all right. So let's come back then just briefly to the big picture. So I'm going to start here where we look at the course of viral infection. So by the time you see the virus, there's this latent period where you have 2 to 7 days before a person gets sick, right? And again, from the time they start to see symptoms, there might be a week before they see more severe disease. And then again, a short time between the time that the disease starts to get serious in terms of lung function before it becomes actually respiratory distress. And then you can get systemic shock and all of the things that would lead to patients dying, right? So the idea is that you would be able to interfere with different processes either in the virus or the host at these different stages. That's why understanding the biology of of the virus and its interactions with us is important, right? So here at these early stages where the virus is starting to get in and starting to get reproduced, we're thinking that this is the time to use those antiviral drugs that interfere with viral reproduction, right? Mm-hmm. This is also the time to use drugs that would turn up the immune system, that would actually prevent the loss of some of the immune components, such as T cells, that would allow the body to respond effectively to the virus and get rid of the virus. But where you see is when that will work for patients who don't get the severe disease. Patients who get the severe disease, there's a second process where you can see that there's a massive inflammatory response, again, generated by our immune system. So this is what you want to prevent with those drugs that prevent the cytokine storm. So things like the anti-inflammatory drugs like the IL-6 blockers that we talked about. This is where you also see the problems with drugs that can, or the problem with the virus, which is it causes coagulation. So treating patients with anticoagulants that are fairly commonly used for other things might be useful.
Right? I think this is probably where a little bit of the counterintuitive understanding of the therapies come from in the media, in the general media. The immune component of it actually can be activated in order to have a better response, but also needs to be tamed down when you have this cytokine storm later on. So you really need to treat with different drugs at different moments and dose it. And that's why you need so many different tools in your toolkit.
And that's why it's so critical that we're buying time to kind of understand where that switch occurs, right? When it's good to boost the immune system versus where it's good to dampen the immune system, right? That's what we've learned just since the lockdown, essentially, right? So is that when I remember reading papers at the beginning saying that they didn't have any idea about whether anti-inflammatories are good or bad. People were being told not to take ibuprofen because it's an anti-inflammatory drug, right? But again, now we understand that, you know, if you have the mild disease, it's probably all right. Or it's probably not good to take anti-inflammatories, right? Because you want that immune response. But if you have the severe disease, then you want to turn down the immune system, right? So we need to understand this actual switch better, and that the loss of the immune response and the upregulation of a related but slightly distinct inflammatory response, which are often treated by the same drugs, we need to understand where that inflection point is. And so we need three different things then, right? We need antivirals that actually deal with viral reproduction itself, and we need some way to either boost or dampen the immune system depending on the point in the disease where we are. And I think that's basically all I had to say, Thomas.
Yeah. So I have to thank you, Terry, for this, I think, a very comprehensive overview of the therapeutics targeting COVID-19. It was very insightful. And I think one thing that's recurring here is that developing drugs is not just a matter of lab work. There's a whole ecosystem involved, going from socioeconomics to politics to, you mentioned also the importance of what's happening at the clinics, educating back the therapies, but also the importance of really understanding the disease in order to actually come up with new therapies and identifying what might win and why it wins. Because we should learn lessons from this infection, but we should remember what we've learned from that infection, because there will be more infections from coronavirus, but also from other viruses out there. So I think it's probably one thing that overall comes out is you need more than one approach here. You need really the whole picture approach against this, and really how important it is to act fast and have solutions that are applicable now, and in the same time work on something that prevents as well, just like vaccines.
And you know, this is where we see also that you know, we need governments that function, right? We need governments that are able to manufacture things like masks, or like we need capacity locally, and we need local government, and we need leadership to deal with these things, right? You could think of this as an opportunity to learn how to prepare for the next one, and I hope that's what we do, because the next one might not might be worse, right? So we just don't know.
Yeah, it's
And
there's been quite a few already coronavirus. It's not going to stop. So in the same time, I know that Terry has prepared for the audience here a series of references that are credible, really high quality references. If you want to learn more about the development of new drugs, the repurposing of drugs, and of course prevention using a vaccine, but also different levels there of detail. Some of them are more summaries, where others are more for the seasoned scientists. So we know that there are people out there are not necessarily all researchers, and sometimes having something that is amenable to everyone and still be of high quality information is appreciated by the community as a whole. So thank you very much, Terry, for picking those lectures. They will be added at the bottom of the YouTube channel, so you can refer to it. So again, thank you very much, Terry, for taking the time to do this. I know you're busy, and it's really appreciated for you to come here and talk to us about it.
it's my pleasure. Thank you.
Thanks, Terry.